Hello, and welcome to A Vision of the Future, Advancing Gene Therapies to Treat Eye Diseases. I'm Alex Nixon with the Center for Connected Medicine at UPMC, and I'm going to take a minute to go over a couple of housekeeping items before handing off to our moderator for today's discussion. I'm excited for today's expert panel on gene therapy and eye diseases, which the center organized in partnership with the Institute for Precision Medicine. This presentation is part of the center's ongoing programming focused on scientific and medical innovation. If you're not familiar, the Institute for Precision Medicine is a collaboration between UPMC and the University of Pittsburgh, and that's dedicated to facilitating the movement of biomedical research into personalized clinical care. And we're grateful for the partnership with the IPM. One final thing to mention is we encourage our audience to participate in the discussion by submitting questions for our panel of experts. We've reserved time at the end to take your questions, and you can submit them by using the Q&A box on the right-hand side of the screen. With that, I'll hand it over to Dr. Alex Anatakis, a UPMC surgeon and physician advisor to UPMC Enterprises, who will lead the discussion and will introduce the other panelists. Dr. Alex?
Thank you, Alex. Appreciate that. As the other Alex has said, my name is Alexander Anatakis. I'm a vitreoretinal surgeon here at UPMC. Welcome all of you online that took the time to spend some time with us today and learn more about gene therapy and eye diseases. As a vitreoretinal surgeon, I'm obviously very passionate about this subject, and thank you all for joining us today. And we're looking forward to a great discussion today. I'm fortunate enough to be joined now by two panelists here with us today who have been in my life in different ways in the past and now in the present, of course. And I'm going to take that just a moment here to introduce them. But we are very lucky to have both of them here today. First, I'm going to introduce Dr. Rob Lin. He is a scientist and a businessman and brilliant at both. I was fortunate enough to work under his tutelage at UPMC Enterprises when I first joined that team in 2018 and worked under his tutelage for many years there. Rob was integral in starting up one of our startups in our venture creation that we were able to do through Enterprises in a company known as Avista Therapeutics, as you see on his swag that he's wearing with us today. He served first as the interim CEO, but then loved his job so much there that he decided he wanted to go full-time as the full-time CEO and now functions as the CEO of Avista Therapeutics. So Enterprises' loss was Avista's gain. So we're very glad to have him in that role, though, guiding our portfolio company. And so I'm going to let him continue his intro for himself here.
Thanks, Alex. Great to see you again. I'm very glad that the audience is tuning into us instead of March Madness, but I guess the people who are going to watch it later recorded may have been able to catch both. As Alex said, I have a background as a scientist. I did a PhD in genetics at Harvard University, and then went on to do corporate and business development at a biotech called Millennium Pharmaceuticals. After that, I was with the Bill and Melinda Gates Foundation, where I ran the finance and strategy for their global health program. And then prior to this, as Alex mentioned, I was working with him at UPMC Enterprises, where we started and funded a number of companies. And as Alex mentioned, went on to go ahead and lead Avista Therapeutics, one of the companies that we ended up starting out of some technology out of the University of Pittsburgh.
Thanks, Rob. And so our second panelist here with us today is Dr. Dan Chung. Dan and I's story goes way back. It's quite an interesting story, at least to me anyways. I was a fledgling medical student looking at ophthalmology in 1999, and Dan was a young resident physician in ophthalmology at the time, and we were together in Akron, Ohio, and I got a taste of ophthalmology from Dr. Chung and helped spur my interest in ophthalmology. Fast forward the clock, about 22 years later, one of our investments for UPMC Enterprises is an exciting biotech, Sparing Vision. And Dan is now serving as the Chief Medical Officer for Sparing Vision, and they are an exciting biotech in the gene therapy space in ophthalmology, hence his joining us today with his vast experience in that area. Dr. Chung was integral in bringing Luxturna to the market. For those that may not be familiar with that, that was the very first gene therapy ever approved in humans, period, in the United States, let alone ophthalmology. and it's still the only gene therapy in ophthalmology that is FDA approved. And Dan was integral in bringing that to the market, and I'm gonna let him tell a little bit more about that story. So it's great to reconnect with Dan 22 years later, and I'm fortunate enough to get to work with him through our enterprises / Sparing Vision connection. And I'm gonna pass the mic to Dr. Chung.
Well, Alex, thanks so much for that intro, and thanks for the kind words of being a young resident. I was probably the oldest resident that they had. So, but no, it was great to reconnect, and it was a surprise when I saw you. I knew you from somewhere. And it took me a minute to figure out where, but and as Alex said, so I've been in the retinal gene therapy space for a while. I did a fellowship at the National Eye Institute in Retinal Gene Therapy back in 1994. We were still using adenovirus and a lot of different things there. And then I spent some time, obviously, at University of Pennsylvania. Was very fortunate to work with, obviously, Dr. Bennett, Gene Bennett, and Dr. Al McGuire there that led the whole Luxturna endeavor there at University of Pennsylvania Children's Hospital. Then went on to Spark Therapeutics and really learned about what it meant to bring a drug to market and payers and things of that nature. So it's really been something very fortunate. I've been very privileged to see kind of a drug going from the bench all the way really to the patient. And we're just hoping that we can do more and bring that to more patients otherwise. Now I'm at Sparing Vision. We are a genomics medicine company based in Paris, France, with also US HQ in Philadelphia, where I am currently. And we're working on gene agnostic ways to hopefully preserve cone function or central vision function in retinitis pigmentosa. And then a second product that we are working on and hope to be in the clinic shortly is something that maybe will hope to restore central vision in patients. And those are all just the starting ideas, and then we'll go on from there with many more applications and possibly into dry AMD and things of that nature. So, but thank you again for the invitation. It's great to be with you all. And Rob, of course, we have known each other for a bit, so great to be with you as well. So look forward to the conversation.
I think we got a great group here, and I look forward to it as well. Dan, you mentioned Dr. Jean Bennett and the work that you were able to do with her, and I think that's a good segue to starting to talk about gene therapy in general, let alone gene therapy in ophthalmology. And maybe it'd be nice if you could walk the audience members through the evolution of how gene therapy in ophthalmology came about. I think we have maybe a mixed audience online, some with probably a little bit more ophthalmologic experience and some with less. And so it might be helpful for those online to hear a little bit about the history of how gene therapy in ophthalmology evolved.
Sure. Kind of in a longer nutshell, I guess, is that ophthalmology is a great field for scientific exploration, and especially in genetics. As you may know, there are over 300 different genes for inherited retinal diseases. There have been genes that have been identified as predisposing factors for AMD as well. And so this has really just been a rich bed for research. And one of those things that was obviously in gene therapy was the idea of, can you really just put back a functional copy of the gene that has variants in it that's causing disease, and seeing basically if you can change the natural history of disease. And obviously the work that was done at the University of Pennsylvania with Gene Bennett and the whole group there with Al McGuire, who was the surgeon there, they really launched the whole idea of doing gene replacement, and the idea that you can put this in a patient, subretinal injection, and have a restoration of function, which we saw in many of the Luxturna patients that were in the clinical trial. And then obviously it went on to commercialization. It's in 40 different countries around the world with Spark's leadership in the regulatory and in commercialization of that, along with Novartis, was the ex-US partner. I think from that, it really just exploded right back there in the late 2018 or so, and where a lot of new companies came out with a lot of new ideas for gene replacement therapies. And I think there was a lot of good research that's still going on right now, a lot of good clinical development folks, and a lot of expertise. But I do think that in that time span, because it's been 7 years since Luxturna was approved, we really haven't had another approval for inherited retinal disease. And I think that just really is an example of how it's not quite as easy as putting the gene back in. There's so many other things that you have to do in terms of regulatory and trial design, vector design, preclinical work, everything about it, there's always somewhat of a challenge. And I think for many of us, this is kind of where we're committed. We're hoping to have more therapies in the future. There are a lot of other companies who are close. So we're hoping, but it really is kind of blossomed. And now there's a little bit of, because of some of the challenges, there is a little bit of apprehension sometimes with fundraising and things like that. But there's still a lot moving forward. So we want to do something obviously more than just Luxturna and get the field and another therapy for patients who there's a high unmet need in the inherited retinal disease space.
Absolutely.
So we need to get something for these folks.
Fantastic. Well, thank you for that, Dan. I think that is really helpful for folks on the line to hear the evolution of what it took to get that Luxturna really up and running into patients. It's a nice segue to thinking about what hurdles are out there for delivering gene therapy in ophthalmology. You touched on them briefly, but I'm going to ask Dr. Lin here to expand on that a little bit more. We know that there are hurdles to delivering gene therapy in ophthalmology. And the things that I'd maybe like to touch on are the delivery modalities, whether it's subretinal, intravitreal, suprachoroidal. Maybe you can walk the audience through that. Think about the inflammation risk, capsids, and I would like you to put some plugs in there for Avista with that. And then maybe talking even a little bit about the patient populations and how that can be a challenge in itself.
Great. Thanks, Alex. Well, there's an old saying that three main challenges in gene therapy are delivery, and then delivery. I think we've taken that to heart at Avista and addressing some of the challenges in delivery. As you mentioned, there are a number of ways to be able to get therapies into the eye. And I think what we found with gene therapy and what Peter Marks at FDA has said that he really likes about gene therapy is it's a very exquisitely targeted therapy because especially in a lot of the inherited retinal disorders, we know exactly the gene that is malfunctioning and we know how to fix it and correct it by the demonstration that Dan and his colleagues showed with RP65, that you can just add back a very corrective gene with the correct sequence, and that'll help cure the disease. The challenge has really been, as you mentioned, in the delivery portion. Dan and his colleagues demonstrated that Luxturna can be delivered through subretinal surgery, which I think is really terrific in terms of being able to address photoreceptors directly. It is a bit of a challenging surgery, and I think is a bit limited in terms of widespread things. So in terms of rare diseases, I think it'll work really well in some rare diseases, but maybe limited to a disease that has a larger population you want to treat. And I think that's where the focus at Avista has been, is really trying to focus on the intravitreal route of delivery. That is a more commonly used for other drugs besides gene therapy. I think a lot of the wet AMD antibodies are delivered in this method, and it can be an in-office procedure that is relatively quick and easy and hopefully relatively painless for the patient. And so I think that's
when I do
it. Especially when Dr. Alex does it. But I think naturally occurring viruses have a hard time crossing the barriers from the vitreous into the retinal cells. And so I think that's one of the things that we're doing at Avista, is trying to engineer through targeted mutations and insertions into the capsid sequence to make those more valid vectors to be able to use for gene therapy in the retina. And I think what we're really excited about the work we're doing at Avista is I think we have found some really interesting capsid variants that we think are best in class and really can push forward the delivery route of intravitreal gene delivery that probably has not been as valid in the past. And so really excited about the work that we're doing there. You mentioned inflammation. That's been one of the main issues plaguing intravitreal injections of gene therapies. And our feeling is that with the improved power of our capsid variants, that we can actually reduce the dose necessary to treat patients. And I think that is a really good route to minimizing the inflammatory issues there. You also mentioned a suprachoroidal route, which is more of a novel route of delivery. We're really interested in that, as we describe that as going around the eye through the back portion into the suprachoroidal space and coming back through the eye through the RPE cells in the back there, and the retinal pigmentary cells there, and so epithelium to come in the back way, and really trying to explore that as well as a separate method of delivery. And so I think real advances in that space, and real exciting about where that can go in the future, and Visa's contributions to that.
Fantastic. Thank you, Rob. I appreciate that. That was really helpful to hear your perspective, and as well as how Avista is trying to solve some of those major problems that we have in gene therapy. One of the major problems with gene therapy is these small patient populations. I think it's important for the audience to understand what we're dealing with here, that inherited retinal disorders, which most gene therapies currently are aimed towards targeting, have an incidence level of about 1 in 4,000 or so. So pretty rare overall. And then once you start diving into that further, you realize that you may have a couple hundred patients for each specific gene defect in terms of being able to address with one therapeutic. To get over that hurdle, one of the things that people are looking at are gene agnostic therapeutics, such as what Sparing Vision is doing with Dr. Chung leading the way as their CMO. And so what I'd like to do for Dan, and direct this question to Dan, is talk a little bit to the audience and give them an understanding of what are the pros and what are the cons of doing a gene agnostic approach that may be able to treat thousands of patients instead of a couple hundred, versus the pros and cons of a gene specific therapy in ophthalmology, and what those two different approaches mean, and how that could look in the future.
Yeah, obviously somebody that has a lot of experience with the whole pathway of a rare disease with RP65, which obviously has a relatively limited population. The idea is that there are just not enough resources and time to address all 300 inherited retinal genes. And some of them are so rare that it's difficult to bring resources in to put that through a clinical development pattern. And so this is when you start thinking about gene-independent approaches. And that's where Sparing Vision, where our lead program is based on a neurotrophic factor that was identified by one of our founders, Professor Jose Sahel, who happens to be the chair of ophthalmology at the University of Pittsburgh Medical Center. So, but it basically is looking at an innate pathway, something that happens in all of us, where we found a relationship between these rod and cone cells. These are light-sensing cells, and they have different properties. The rods are in the periphery, and they're more for nighttime vision and dim light side vision, whereas the cones are more centrally located and concentrated. And that's what most people are using right now to look at this screen, are these cones for daytime vision, color vision, and your best acuity. And the idea is the question was, why is it sequential? We know that most of these genes affect something in the rod photoreceptor physiology. So we know why those go down and why they degenerate. But there didn't seem to be a real direct link genetically to the cones. And so they found a factor, and they termed it rod-derived cone viability factor, and simply something that there are two forms. One is secreted by the rods, and it binds to the cone photoreceptors and goes through all these pathways to support cone health. And there's also a big antioxidant factor that's found, and those put together is how the eye keeps the cone cells healthy. And the idea is, well, on a gene-independent level, it didn't really matter why the rods died and the RDCVF wasn't produced. It's simply that the rods are no longer there to produce the RDCVF. And so can we just put that RDCVF back in the eye and at least hopefully slow down the disease or stop the disease from going any further? And it addressed a large class of inherited retinal diseases known as retinitis pigmentosa. And these are known as rod cone dystrophies. And really any mechanism or the pattern of disease where you have rods die first and then cones, this would be applicable for. So this is where we're now in clinical trials right now. We're in our first part in a dose escalation. But there's a lot of other gene agnostic modalities where they're not targeting a specific gene. And this is by no means saying that we shouldn't pursue those. There's still a lot of great work going on for X-linked RP and RPGR. There were some programs in choroideremia and things of that nature. So the idea is, how many shots on goal can we get? The more things we try, the higher the percentage that something is going to be successful in the end. And so gene agnostic goes for a lot of different things. It could be something like optogenetics, which we've heard a lot about. And that's obviously not dependent on the genetic variant. And stem cells also, that's another way of going gene independent. So in my opinion, it's good that we are in both realms, the gene specific as well as the gene agnostic. Because one thing about the gene agnostic, the pro about that is that it will help us to address patient populations that are really, really ultra rare. The biggest populations in ophthalmology, if you go into Stargardt's or something, which a lot of companies are going after because it's much more prevalent, you're talking potentially thousands of patients that way. But then other genes you look at, and there may be 100 in the whole world. So it really gives us that option. I do think that, and I'll stop after this, is that a lot of times the gene specific can be, in terms of Luxturna, it really does, in many patients, obviously not in all patients, can have a significant recovery of vision, whereas we're trying to slow the progression of disease. So it's definitely a give and take.
And if I can just follow on that, Alex, I think it's,
Yes,
it's really, really complementary. And, I think the instances where there are treatments that are available for gene specific, I think, as a patient, that may be one of the better opportunities for you, but there's always going to be a number that there's just not enough of a patient population to be able to either run the trials or be able to create a therapeutic for it. And I think that's where the work that Dan and Sparing Vision are doing is really complementary to that. And so I think it's a matter of both. And if you can do one, that's great. And if not, then I think the gene agnostic approach for Dan and Sparing Vision will help pick up those people.
Yeah, thank you for adding that, Rob. Appreciate that. Appreciate it. And again, I'm directing questions to one person or the other, but please feel free, both of you, to jump in and give your thoughts on things as well. And I'm going to get back to the cell therapy part of things that you had mentioned, Dan, and I'm going to direct that one towards Rob. But you also had mentioned optogenetics, and that's an exciting field in ophthalmology, and there's a couple different players that are out there making some nice advancements there, and one of which is Sparing Vision. And so I think it might be helpful for the audience to hear a little bit more about what optogenetics means and what that can mean for patients along the disease pathway when they're no longer eligible for certain therapeutics with, quote, traditional gene therapy.
Yeah, I'm happy to start the optogenetics. So optogenetics traditionally targets a different cell population, whereas in gene therapy, we're targeting RPE cells or photoreceptors. Most of the times, optogenetics will target more the inner retinal cells, ganglion cells, the bipolar cells, even Mueller cells. And it's the idea of enhancing these cells to be more light sensitive, because intrinsically, or for the most part, the light sensing cells that take the bulk of the responsibility are the photoreceptors. But these inner retinal cells play a role in that to a smaller degree, and it's basically trying to enhance that. And a lot of times the idea is, can we take patients who are very, very degenerated, that have little or no function, and try and bring them back with this pathway? And the idea, again, is gene independent, because you're targeting a different pathway than the actual gene variant that is causing the issue. And there's a lot of good work going on in that. Now, for us, we get compared a lot to optogenetics in the sense that we're trying to do the same and take patients who have very little function and try to restore function in that. But instead of targeting the inner retinal cells, as I mentioned, we're still targeting photoreceptors. But these photoreceptors are what we call dormant cells. Basically, they've lost that ability of what we call phototransduction, which is the ability to take the light that's coming into the eye, convert it into an electrical impulse, and send it to the brain, because the brain is what sees. The eye is just this really complex relay system. And so the idea is we put in a G protein inwardly rectifying potassium channel, G and GERK, and the idea is that it will now produce a truncated form of that phototransduction in the cone cell itself, hopefully restoring functional vision. So I do think that optogenetics and this other form that we're working on could play a role in restoring vision. How much vision, I think, is still something that we are still trying to figure out. And obviously, once we get into clinical development, we'll know a lot better on that.
Fantastic. Thank you, Dan, for sharing that and sharing his vision's approach to it, which is, a little different, right? But thank you for sharing that with us. I appreciate that. And I'm going to take just a quick pause here to, I know it got mentioned in the chat by Alex, but I'd also like to take a moment here to please encourage the audience to put questions in the question and answer box. And when we're going to definitely reserve some time at the end to answer those questions. So please feel free to add them there, and we will try to collate them as much as possible and get to every single question that we possibly can. So again, I would encourage you to do that. So I'm going to pivot here towards Dr. Lin here and want to hear about your thoughts on cell therapy in ophthalmology in terms of a different way of approaching things instead of a traditional gene therapy, more of a cell therapy. And one of Dr. Lin's expertises as well, or experiences in the past, has been with one of our other startups, Blue Sphere Bio, which is a cell therapy-based approach for oncology. So a little bit different, but Dr. Lin is so brilliant that I know that the lessons learned there can maybe potentially apply to his thoughts on ophthalmology cell therapy. So I'd love to hear your thoughts on how you think cell therapy might be applicable in ophthalmology, and maybe not the near future, but maybe more like distant future, and the challenges that go along with cell therapy.
Yeah, no, I think it's a very intriguing concept as opposed to delivery. The main issues with cell therapy are more in the manufacturing side of things. And so, as you mentioned, I'm on the board of Blue Sphere Bio, which is a cell therapy company that's working on cancer. And I go through many days of discussion about the challenges of manufacturing, but it's really important because at some point the manufacturing process is your product there, and so getting the right cells with the right properties to grow in the number and the quality that you want is perennially a challenge there. But it's probably beyond the scope of today's discussion. But when we think about specifically cell therapy in the eye, I think there are a number of avenues that are really interesting. And I know, Alex, you and I look at a number of companies in the past that have different strategies with this. In terms of an intravitreal approach, the cells are too big to cross the barriers into the retina. So generally, there are some approaches where you can inject some cells and hope that they make the right proteins or growth factors or whatever you would like, and have those factors pass through the cell barriers into the retina. And so I think there are some people doing some interesting work on that. I'm not seeing any real clinical data on that, but it's a really intriguing concept. The ones that have been in clinical trials have involved injecting cells into the retina through an injection in very similar to the subretinal injection that Luxturna is delivered by. And I'm a little bit probably less excited by those because you're injecting, in the case of Luxturna, a liquid into a bubble that you're creating in the retina, and that typically dissipates, and the gene therapies do their job there. But when you inject cellular mass, they don't have any place to go. So you're pretty much creating a bubble of extra cell materials, and that may be what you want, but it is really challenging to figure out the right type of cells you want to inject, and then the right amount that you don't inject enough of a mass that you end up creating a retinal detachment. So I think those are particularly challenging. And then I think another one that we looked at quite a bit is artificial cellular therapies that create an artificial RPE layer. And so if you think of this as a TARP that keeps the eye immune privileged, if there's damage to the RPE, can you create almost a patch that you can put over any gaps in the RPE? And I think that has some really interesting scientific progression. Again, limited clinical data there, but thinking about how you can create an RPE, artificial RPE, whether it's cellular or some other method that can help patch those gaps. I think there's some promise there, but again, particularly challenging, especially since the RPE cells are polarized. So one side is very different from the other side of the RPE. So you not only have to get them in, but get them to maintain that polarity in the cell type as well.
Yeah, the orientation is so critical with those, and there has been some interesting approaches I've seen where you use artificial matrices to have the cells already growing on the little wafer, so to speak. But and then you have something artificial in the eye too. So then there's challenges there. And at the end of the day, it's the retina is still an electrical system, of course. And so to your point about the cell therapy, if you think about injecting photoreceptors, they have to wire themselves correctly, and that's quite a difficult challenge to ask of those cells. And so it is interesting field, but a lot of hurdles to get over here, I think.
Yeah, there's one other thing I did want to mention. There is a cell therapy with that does go intravitreal, and it's the idea that it's emitting a lot of different protective factors, and that I believe has had some favorable early results, and I believe they're going on to pivotal trials. I do think it's important also to mention that cell therapy has gotten some negative publicity in the last few years because there are those entities that are basically advertising cell therapy and basically charging patients. And the idea is that not everything, if you go on clinicaltrials.gov, which lists clinical trials, just because it's on there doesn't mean that it has gone through the FDA approval process, the whole investigational new drug application and things of that nature that we in industry go through. And we always let folks know they should be very wary of any entity promoting cell therapy that charges you thousands and thousands of dollars. And you should really speak to your healthcare provider about those types of entities. And that has received a lot of negative press, and patients have been injured by that. So just be very careful when it comes to those type of cell therapies, because if it is a well thought out and approved FDA trial, that's the kind of trials you want to be a part of.
Yeah, it's unfortunate that there are people out there that'll prey on the desperate, of course, and people
Right.
losing vision are desperate, and rightfully so, and understandably so. It's one of the scariest things in the world to lose vision, and the fact that there are people out there that unfortunately prey on that is disconcerting. And so I will echo Dr. Chung's point that if you're thinking about something along cell therapy, make sure it's very well researched before you make any leaps. And we're always happy to see folks here at UPMC Retina and give you some guidance as well. So put a little plug in for our department, right? So moving on to, we've discussed a lot about gene therapy and gene replacement and how that's applicable for autosomal recessive inherited retinal disorders, and how that can be really successful, a la Spark and Luxturna. What we haven't talked about too much yet is more autosomal dominant retinal dystrophies. And the difference there, of course, is that now instead of a lack of a protein, you have a potentially toxic protein. And so just replacing it with a healthy copy isn't always enough. And so I'm going to direct this to both of you. I'd love to hear what your thoughts are on gene silencing and gene editing, and how those things may be applicable to ophthalmology gene therapy in the future. And whichever one would like to go first, I welcome that.
Well, I think I'll hop right in. I think really exciting work with some of the CRISPR technologies that have been out there, and the capabilities of them. I think most tested ones thus far are mostly in the slice and dice phase, as I would like to call it. So they're really good at cutting genes and making sure that they don't function. And so a lot of this work has been directed toward the liver, where the lipid nanoparticles are easy to get to. And I would assume you could do something similar in the eye. Again, I'm going to harken back to delivery as being a real challenge here, to make sure you can get your CRISPR enzymes and the guide RNAs to the right place, so you're cutting the right protein. And in this case, you want to cut the misformed or mutated one without damaging the normal one. So it's got to be very precise in terms of where it cuts. But really excited about some of those technologies, especially on the autosomal dominant side. And then along with that, I think a step in complexity beyond that, beyond just the slice and dice approach, is that you can use the CRISPR enzyme if you sort of take out the cutting function of it and use it just as a guide function, where you can attach some transcriptional activators, or in this case, perhaps a transcriptional repressor to it, and then have it targeted to the right place to help silence those genes. And so a lot of this is still being developed, but we'll still need delivery to the right cell types to be able to enact that. And I think huge advances there, both in terms of being able to create the right set of CRISPR type enzymes to do it, and being able to guide them to the right place. So I'm excited to see what comes in the future there.
Yeah, and there's definitely work on gene therapy on this realm as well, where the idea is that you actually put in two different entities, one that is actually going to knock down the gain of function, or the mutated protein, and then comes in with a replacement. And sometimes, depending upon the disease, you may not even need to bring in a replacement. You just need to knock down and the allele. So we know we have two alleles, the affected allele, and that the copy that doesn't have the defect will be able to produce the protein in enough that it can alleviate the disease that way. So it really is somewhat a little specific. The one thing about we go from the realm of gene independent to gene specific, and then you have variant specific. So you have to think about those types of things as well, because obviously the pond gets, or the funnel gets smaller and smaller as you go, and that can also have some challenges as well. So
Thank you both for that. That's very helpful to hear your thoughts on that. So we focused, again, mostly on what we've talked about so far has been orphan indications, mostly inherited retinal disorders, and how gene therapy can be applied to those. Gene therapy is also being looked at now for much broader patient populations for diseases that are much more prevalent, such as diabetic retinopathy, even age-related macular degeneration, and specifically some companies out there doing it well, Regenexx Bio, Adverum, 4DMT. I'd love to hear your thoughts on comparing and contrasting what it's like developing a gene therapy for a rare disease versus developing a gene therapy for a more prevalent disease. And I'm going to let Dan go first on this one.
All right. Well, one of the things is it's probably a little easier to get resources for a prevalent disease. And the idea is that you'll have a large population to address. The idea that delivery, going back to Rob's point, delivery here, that it's one thing to be able to do a couple hundred subretinal injections a year, but it's different when you think you're going to do thousands and thousands of subretinal injections. I'm not sure that's necessarily possible. I guess it depends on how effective the drug is. So the idea that gene therapy can be used, and it's really here, is to alleviate the burden of the repeated anti-VEGF injections to keep wet AMD in check. And so that's part of it. The idea is really about improving quality of life by not bringing patients in over and over again. And as you mentioned, there's several companies that are looking into that. So so that's, to me, is one of the differences. But even gene agnostics, we are also looking at the application of dry AMD because dry AMD, one of the first things that is affected are the parafoveal rods. And so we know there's a deficit of rod function there, and probably a deficit of RDCVF as well. So I do think that AMD is one of those things that many people are looking at to really bring a product that might have a slightly different mechanism of action, or to simply use what is the anti-VEGFs and put it into a gene therapy. So maybe it is the one and done kind of thing. So,
and then I think further on those points there, I completely agree with Dan that that one of the reasons that we're really interested in intravitreal delivery is being able to increase the population that's addressable. And so when you're talking about the more prevalent diseases in the eye, I think that's really the direction that we need to go. One thing I'll point out, which is something that's been on my mind quite recently, is that manufacturing constraints are a lot different when you're looking at producing hundreds or thousands of doses for some rare diseases versus hopefully hundreds of thousands and maybe even a million type doses for a disease as prevalent as wet AMD. And so thinking about that manufacturing scale, it's something that we've had discussions with Avista's partner, Roche, as they're thinking about some of the larger markets as to how you can increase manufacturing capacity and capability and scale up at a very early stage to be able to address the larger populations.
And Rob, I think it's a nice segue to something else that we need to think about. It's unfortunate that it's real life, but all this stuff has to get paid for. And getting gene therapy paid for with the rare disease sometimes is a little easier because insurers and the payers understand that there's only a few patients that need this versus the more prevalent diseases. And I'd love to hear your thoughts on pricing and gene therapy and ways that we could maybe solve that part of the problem.
Yeah, so I think people have seen some of the prices of gene therapies, and they've been extraordinarily high. And I think a good chunk of this is coming down to the cost structure to make gene therapies, to manufacture gene therapies, especially for a small rare disease population. Both the manufacturing process that needs to go through after you reach approval, but all the different investments to do the scale up and purification of the process throughout the way. And so I think that's been a real challenge. I have hope for the future, though. I think a couple things stand out for me. One is that I think in cell therapy, the CAR T manufacturers have managed to do a really good job in driving down the cost structure as the scale grows. And I would assume that something like that could happen in the gene therapy space, especially if there were a product that was approved for a large population like wet AMD or dry AMD, as we've discussed. I think there would be a lot of improvements there driven by the scale. And again, thinking of that early, I think is a really good part for those more prevalent diseases. On the rare disease side, I think a lot of the action is still going into both manufacturing and regulatory harmonization. And so if we can achieve a day where most of a gene therapy is the same and you swap out either the gene of interest or something in terms of a cassette, and FDA and the other regulatory agencies around the world allow a more streamlined and hopefully cheaper process to do the regulatory studies that are necessary to make sure it's safe. But having a large part of the platform be the same, I think will help drive down some of those costs. And so I think I'm optimistic in the future, but right now I think there was just a product that was approved for $4 million list price, and so really dramatic, huge costs, especially on the rare disease side.
Yeah.
So I think Rob covered that really well, Dan. So we only have a few more minutes before we want to open up to the question part of things. So there's one last part I definitely wanted to make sure that the audience members get to hear about, because I know you were so critical in the development of this part of it when it came to Luxturna and its development. When we think about endpoints for trials for approval for inherited retinal disorders, it's different for those than we think of for a lot of things in ophthalmology. A lot of things in ophthalmology, it's pretty simple. Did the visual acuity get better by 2 or 3 lines on your traditional Snellen and eye chart? If it does, it's probably going to get approved. It's different in inherited retinal disorders, and I'd love for the audience to hear your perspective and maybe expand a little bit about how you guys at Luxturna and at Spark were able to overcome that.
Yeah, so I'll address the whole idea of a novel endpoint. The novel endpoint is always relatively risky because the reality is you can make a great novel endpoint, but the regulatory bodies in the end may not believe that it's adequate. So, but for us, we knew that going in that inherited retinal disease, and in this case RP65 disease, that the visual acuity could stay stable for a relatively good period of time. And there's such variability with that. In our natural history study, we found that very young patients could do very well or extremely poorly. But so is the idea really that in working with regulatory agencies who gave us some encouragement that they wanted to see a product or a test that would show improvements in the quality of life, what we call activities of daily living or functional vision, which is different than visual function. And that's really how the mobility test came about, is the idea that patients in that disease, one of the debilitating things was the loss of light sensitivity, and which meant they couldn't do activities at different light levels. The reality is we could see perfectly fine, but if there's no light in the room, we can't see anything. So the idea was, can we measure that? And that's how the mobility test came about, is that we tested them at lower light level. I mean, at one light level. After treatment, we want to know, can they function at a lower light level? And therefore, the extrapolation was then they could do more things, more activities of daily living. Therefore, there was an improvement in the quality of life. So with that said, in ophthalmology, there really are only a handful of endpoints, and most of these come from the glaucoma world or the AMD world. And as you said, visual acuity, you use a special ETDRS chart. It's a very uniform chart, but you need 15-letter improvement when it comes to the FDA. And for the EMA, I think they might consider 10-letter improvement. Well, for inherited retinal diseases, that's a pretty high bar. So a number of pivotal trials have not met that, and unfortunately, their products were not licensed. The other one is microperimetry, which is kind of a pinpoint grid in your retina that determines the sensitivity of that area, and that has a set amount. I think it's what we call 7 decibels and 5 predetermined loci, and that kind of came from the glaucoma world. So we're always looking at other endpoints that might be more applicable for the inherited retinal disease space, such as full field light sensitivity threshold testing, FSTs, low luminance, acuity. So there are a number of other things that we are trying to talk to regulators about, let them know that we believe that these would give us good indications of product therapeutic value. And I think it's just a matter of getting more conversations on that. And hopefully we'll have a few more endpoints that will be accepted in some of these trials coming up.
Fantastic. Well, again, for the benefit of the audience, I commend you on what you were able to do there to come up with a novel endpoint. That is no small task, and the fact that you're able to achieve that was absolutely fantastic. And
well, it's a big team that did that, so
I know it's not alone, but on behalf of you and your team, thank you from the ophthalmology team. So appreciate you doing that. I think we're going to pivot here towards the audience questions here. And I'm going to try to go through these and make sure that we go through all of them. I think we'll have time to probably go through all of them. The first one is, what are your thoughts on other types of carriers for gene therapy in the eye? And they specifically mentioned nanoparticles and other viri. Rob, you want to take that one first?
Sure, I'll give it a whack. I think I know Sven, so hi, Sven. I think there's other methods, as you said, of delivery throughout the body. I think people have tried lipid nanoparticles and some other different types of ways to do it. I think at least in the eye, our thought on delivery, I don't think AAV is perfect, but what AAV allows us to do is have something that's been in humans. We know how to manufacture. We know how the body reacts to small doses of AAV. We also know exactly what happens when you add really high doses of AAV. And so I think, particularly for eye applications, AAV is a tested and tried and true type of delivery vehicle. It, again, is not perfect. And one of the limitations is in terms of the size of the insert and the size of the gene that you can correct with it. But I think there's really good ways that people are trying to think around that concept. There are other methods of delivery in the eye, some of them viral. There are people trying other viruses that might have slightly larger carrying capacities. And again, the trade-off there is switching to a novel platform introduces a lot of different variables in manufacturing and how the body might react to things. So definitely some considerations there. I've seen a couple of non-viral types of things in the eye. I've seen at least one company trying to do entry into cells using electroporation in vivo in the eye, which I don't know, Dr. Alex, you can tell me how your thoughts about that, but having electrical shock to introduce DNA into your cells, especially in vivo in the eye, doesn't sound like something that I want to sign up for, but I'll let you comment on that one.
I think if it worked well enough, I know which company you're referring to. I think we could convince patients to take that on. I do think it's not quite so painful, but The unfortunate truth is the efficacy of that methodology seems to be a little less than we were hoping from what the early trial results that I've seen anyways.
With non-viral, what we found is that the positives is that they're relatively low cost, low immune response, and you can put a pretty big package in them. The downsides, or the things that we're still working on, I think, is the idea that they had low transfection efficiency. I think that's being somewhat fixed now, so that's definitely better. I think the biggest thing is durability, and that's something that I think still needs to be addressed to some point. And when it comes to the other viruses, obviously AAV is the majority of what we use right now, and the other viruses, as Rob mentioned, can be used for bigger packages that we can insert. The biggest thing is also how they incorporate. AAV is usually set up as a little episome, so they don't really incorporate into the host genome. Whereas some of the other viruses, that has been one of the historical issues with those. And I think those two are now being mitigated with some of that risk being taken away. So again, as Rob said, delivery. So.
Yeah, the safety, though, to your point about the other viri behaving a little bit differently than AAV is so critical. First, we got to be safe first, and then we worry about efficacy second. If we don't have a safe therapeutic, it doesn't matter. So I'm going to paraphrase the next one, and I'm going to direct it towards Dan. The question was, are the therapies being developed in the pediatric population? And the answer is yes. A lot of times in these hereditary retinal disorders, they start relatively young, and so by definition, you need to go into the pediatric population. And so I'm going to paraphrase it and change it up just a little bit. And Dan, maybe speak to the challenges of working with a pediatric population in gene therapy and ophthalmology.
Sure. I mean, Luxturna, although it's able to treat adults as well, the indication is actually for patients who are 12 months and older, who have two biallelic variants in the RP65 gene, and so really it is a childhood disease in nature. The challenges are, number one, the surgeries can be definitely a little more challenging because the pediatric eye is definitely different than the other eye, and doing a vitrectomy and subretinal. And luckily, we have a lot of great pediatric retinal surgeons that are very accomplished in that. The other thing is endpoints. One of the things in Luxturna when we were trying to do that was we had an age limit of 3, but one of the inclusion criteria was that they had to perform on the mobility test. Well, 3-year-olds would try and they would sit down on the course and start playing with the obstacle. So it didn't really.
I have a 3-year-old, so that's why I'm laughing.
So, but we were able to get some 4-year-olds, but there was a lot of other testing too that we had to really think about. Would a young child be able to do this? And even consent, because there's a whole idea that you have to assent the patient who is under 18, not just have parents' approval on that. And there are a number of other things that we have to think about because children are not small adults. And you have to really tailor what you're doing. Same thing with the mobility test. We had to think about how do we design this that a child could do this as well as an adult. So basically physical dimensions and things of that nature. So doing a pediatric trial definitely brings in a whole nother scope of things. And you really have to keep in mind that these are children, not small adults. So
fantastic.
There are a number of other indications. There's another program that's doing another childhood retinopathy. So that the pediatric population definitely has been represented.
Thank you, Dan.
And just to answer the original question, I think, absolutely trying to find the right time to intervene for patients. And so if it's early intervention that helps either treat the disease and keeps the vision going or helps arrest any of the progression of the blindness, I think, we want to be able to treat it at the right time. And as Dan has highlighted, it gets more challenging when you do it in a pediatric population. But if that's where the best chance of intervention succeeding is, that's where we want to go.
Yeah, and just to give a little plug for Avista and their opportunity or their challenge of doing it intravitreally, doing a vitrectomy and doing subretinal delivery in a child is a different process than doing it in an adult. And so if Avista is able to accomplish what they think they can do with intravitreal delivery, that's going to open up a lot more opportunities for pediatric populations to be treated more easily and more safely. The next question was, again, it's a yes or no answer, so I'm going to paraphrase it because we actually have talked about a little bit. and I'm going to direct this one to Rob because I know you've been following this story pretty carefully. So the question was, is gene therapy available for AMD, for age-related macular degeneration? And there's nothing is FDA approved right now, but there are multiple trials ongoing for wet AMD. And I'd like you to expand and give your thoughts on what you've seen with those trials and where you think the future lies for that.
Sure. And there's a segment on dry AMD as well, which I'll leave for Dan since he's got a program that they're working on there. But for wet AMD, there is no currently approved gene therapy. There are a number in clinical trials. As you might imagine, for an indication this big, people moved away from the subretinal delivery to different methods. Most of them in the clinic right now are focused on intravitreal delivery, although Regenix and AbbVie do have a suprachoroidal program that they're working on. I think the results have been generally decent. It depends on what your expectations, I guess, is how I would phrase it. The issue with wet AMD right now is that docs like Alex take really good care of their patients. And if people keep coming back for their anti-VEGF injections, do fairly well on their vision. So you're starting from a baseline of people who are doing generally well, and it's a safe and tolerable treatment. So if you're going to do gene therapy in this space, it has to be absolutely safe. And you want to be able to replicate some of the vision gains people are seeing with the anti-VEGF injections that Alex gives people. So it is a high bar. And I think what we've seen is a drastic reduction in injections needed by people who receive the gene therapy. There have been some inflammatory issues, and I think that's one of the areas where better vectors like the one that Avisa is developing might help in that front. But I think it depends on what the standard for patients and doctors is for a gene therapy. If you want it to be one and done and not ever have to come back to see Dr. Alex, I'm not sure we're there yet. If you just want relief from seeing him once a month and, we'll tolerate coming in once every 3 to 6 months, I think we're getting closer to that level.
Fantastic. Yeah, Dan, if you'd like to chime in on the dry AMD side of it and your thoughts there.
Yeah, I mean, dry AMD side, there's no gene therapy currently involved in that. There are 2 products that have come out. I think that it was a major breakthrough for the dry AMD space. Obviously, it's not perfect. There's basically a rate reduction in the progression of what we call geographic atrophy without any real significant clinical improvements. And I think that's probably the topic of discussion right now. And one of the products has had some side effects, although very, very few patients, but the side effects have been relatively significant. So I do think there is more room for other products in the dry AMD space. And I think that's kind of one of the things that we're looking at is, can we apply this gene agnostic approach with saving cones through our product with dry AMD as well. So I do think there's more work to be done in that. And as you said, Rob said that there's a lot of work on the gene wet AMD side, but we have a lot of good products for that already. So it's really more about patient burden on those things. So I think the dry AMD space is still pretty open for other therapies, and but we're grateful that there's a breakthrough and that these patients have something at this point.
Absolutely. Thank you. So I'm going to take a crack at the last 2, if I can, here for myself. We mentioned about the cost a little bit, and this question was, do you have any plans to help mitigate the costs of these therapies by calculating the cost of going blind? In oncology, this has been the key messaging. And the answer there is yes, of course. And maybe Dan, I'll have you chime in just a moment here relative to Spark and how they came up with their pricing. Absolutely, there's been some evidence that the cost of going blind is approximately $200,000 per year. When you think about the care that person needs, if you think about taking them out of the workforce and all the costs that go along with caring somebody who's visually impaired, those have been factored in when they think about therapeutics that are for diseases that are blinding. And so absolutely positively, that is how the justification for the price tag of Luxturna for $425,000 came about. And I'm sure Dan can expand a little bit more in depth on why they came up with that and how they came up with that. But absolutely, that is something that is being factored into things. But the unfortunate truth is also, at the end of the day, there's a pool of money. There's a pool of money that has to go around for everybody to take care of those folks.
Mhm.
And how we divide those dollars can be challenging at times.
Uh, Yeah, exactly on that. Yeah, we definitely take that into consideration, is the burden that not having full vision will be in terms of loss of productivity. The other thing that a lot of times was not taken into account when we talked to agencies and payers and things was the cost incurred by those caretakers. Remember, for us, there was a lot of pediatric patients here, and there were parents who basically gave up their careers or stopped working or something so they could really help their child full time. And those costs aren't really calculated. It's usually the direct cost, and sometimes we need to look at the indirect cost of what that looks like. And a lot of times this is really based on what they believe durability is. Is this going to last 5 years, 4 years, 10 years, 40 years? And that really impacts the amount of payment, plus the idea of how prevalent it is. And what is it going to be a burden? How high a burden will be onto the healthcare system itself? I think there are so many different factors that go into this. And each jurisdiction has a slightly different way of looking at it. You know, it's kind of the US has this private insurance-based kind of thing. And as Spark, we blanketed the United States with that. And obviously Europe has socialized medicine. So it's a little different. You're dealing with different entities there, and they all have slightly different philosophies. So there's a lot to be put into a pricing modality, I guess, is what you would call it. Yeah.
Fantastic. Well, thank you, Dan. And we are just past the top of the hour, and I want to be respectful of everyone's time. There was one last question that a person had asked specifically to their diagnosis, and in the interest of privacy, I won't say exactly what's happening there, but the person asked about hope for that patient. And my answer would be yes. One of the things that I was trained with research, just hope in science that these great folks like Dr. Lin and Dr. Chung are bringing to the clinic. And so, yes, there's always hope for advancement in the future and ways that we may be able to solve vision-threatening issues in the future. And I thank both of my panelists from the bottom of my heart. Thank you so much. I think this was a fantastic discussion. I think the audience learned quite a bit from both of you today, and I thank you again very, very much for being with us here today.
Yes, thank you very much, Alex, Dan, Rob. Appreciate it. It was a great conversation, and I think we can all leave feeling pretty excited about the future and the potential that folks like you, Dan and Rob, are doing to treat these diseases. And thank you to our audience for your attention. And hope everybody has a great rest of their day.
Thank you all.
Thank you.
Bye-bye.